audience as well. Myself, Anjali from University of Rajasthan. By training, actually, I am a biotechnologist, and recently I am working on nanoparticles, and in collaboration with MIT Jaipur, we are trying to develop a membrane which has anti-fouling properties. When we purify water, then we use certain kind of membranes, and fouling, or especially the biofouling of membranes, it's a complication, a problem. We have to regularly change the membrane, which makes it costly as well. So we are trying to develop membranes so that the fouling property can be reduced using nanoparticles. And one of the case study I will be discussing here, which is on the effect of zinc oxide nanoparticles on biofilm formation in Bacillus subtilis. So for the students, what nanotechnology is, any material ranging between 1 nanometer and 100, which has a diameter between these two sizes, it is nano material. And here in the figure you can see various biomolecules, and you can see the range, and you can have an idea what the small size of nano material is. Now nowadays, why it is important? Since in this figure you can see the host is asking, the answer is nanotechnology, and what is the question? That means for all the questions existing now, nanotechnology can be an answer. And these are few applications. It is used in electronics, medicines, textiles, cosmetics, transportation, and so many endless applications it has in our day-to-day life. We are regularly using the products. Coming to the topic, so can nanotechnology be used to get safe drinking water? This cartoon depicts, and of course the answer is probably yes. So we can get safe drinking water. We can remove the problem of membrane fouling using nanoparticles. Coming to water purification systems, the methods we use. So, three main methods: the particle filtration, and it filters or blocks few bacteria. Then there is sand particles, yeast, so many things which this particle filtration blocks. Ultrafiltration, of course, which is a better method since it blocks a wide range, and the RO which we use in our home, the Kent RO, AquaGuard, Eureka Hooks, the endless. So these RO or the reverse osmosis filters which use membrane, they only allow water molecule, except water molecule, all the molecules are blocked so that we can get purified, safe drinking water. In filters which we use, generally four kinds of membranes are used. Microfiltration membrane having a pore size of 0.1 micron, and it allows viruses, multivalent and monovalent ions together with water. Ultrafiltration, it blocks viruses, bacteria and some solid particles. Nanofilter membrane, it allows only the water molecule and few monovalent ions. And the best one till now, RO membrane, which allows only water, and it has a pore size of around 5 angstrom. Now, what membrane fouling is, which we were discussing earlier. So, this is the new membrane, the new filter which we purchase. Gradually, as the water is filtered, the contaminated water, it has certain sludge molecules, EPS, simple soluble microfluid particles. So gradually the pore size is decreasing, the pores are getting blocked, and finally a thick cake is formed on the pores which make the membrane impermeable, and gradually the water purification is hindered. And this process is called membrane fouling. And this cake formation is due to the formation of biofilm, the EPS secretion by the bacteria, different bacteria which are present in the microbes which are present in the water which we want to get purified. Now membrane fouling, it is in general there are 4 types. colloidal fouling, organic scaling, and biofouling. So in biofouling process, bacteria, they form the biofilms. Now, why this biofouling is important? Is it really bad for membrane systems? Yes, it is. Why? Because first of all, it reduces the membrane flux. It increases the differential pressure and feed pressure. Membrane is degraded. It increases the salt passage, thereby reducing the quality of purified water, and lastly, it increases the energy consumption. Conventional anti-fouling methods are generally used, which are use of chlorine, chloramines, ozone, iodine, hydrogen peroxide, formaldehyde, butyraldehyde, etc. We are focusing on we are trying to develop functionalized membranes using nanoparticles. So for that particular study, we have used zinc oxide nanoparticles, which were synthesized by chemical wet method, and they were characterized using XRD, UV-vis spectrophotometer, and TEM, which says the size was hexagonal and the shape was hexagonal, and the size was on an average around 14 nanometer, which was cleared by XRD also. Now, the next step was to check the antibacterial activity of these nanoparticles on Bacillus subtilis. So we used wet diffusion and disk diffusion method, and we found, after keeping the bacteria overnight using nutrient agar media, that the growth of bacteria was inhibited in the presence of zinc oxide nanoparticles. This was confirmed, and we want to check whether the nanoparticle is killing the bacteria or it is inhibiting the growth of bacteria. So we use 2-difitc and PI, propylium iodide, and this is the normal. Here I think due to the problem of projector, the red color is not that much clear. So this is the control where all green cells are observed, no killing, and then gradually the red cells, these are the cells which are getting killed, the bacterial cells, the killed ones, and the green, they are the live cells. So ZNO nanoparticles, they are inhibiting as well as killing the bacterial cells at different concentrations. This is the cell image. In control, we can clearly see a good amount of bacteria, but when they are treated with ZNO nanoparticles, the shape and the size of bacteria is getting changed. They are clumped, their membrane is damaged, and they are round in shape as compared to the control one. Now coming to check the biofilm activity of bacteria, we incubated the bacteria. These are the control, and this is the broth with nanoparticle. After slide, we can see that there was a linear chain formation, which is the initial stage of biofilm formation in treated one, whereas the number of cells are higher. Here the number of cells are reduced, and you can see the biofilm as well. On further magnification, we can see the round cells, the damaged cells, the membrane is damaged, deformed, and here also similar results. And we can see the nanoparticles also, which are adhered to the Bacillus cells. Now, this ZNO nanoparticle, why it is inhibiting or killing the cells? So, some oxidative stress it may be inducing in the bacterial cell, due to which the cells are not normal. So, in order to evaluate the oxidative stress, we use Catalase Assay. The control one, this is before incubation. And then at the end we can see together with catalase, what catalase does, when we add hydrogen peroxide, it releases oxygen, and this oxygen was made to bind with Triton X100. So this Triton X100, it captures the oxygen released by the H2O, and this layer of foam formed, it is the control one, a thick layer, that means high level of catalase activity, and gradually this foam is getting reduced clearly indicates that catalase activity is also getting inhibited. Now at the end, I would like to conclude that ZNO nanoparticles, they are affecting the activity of biofilm in Bacillus. Around 86% production was observed, morphology was changed, and now based on these results, we are about to fabricate membranes with nanoparticles of ZNO and we will check the biofouling activity. I would at the end acknowledge my students Pooja and Arpita, Soft Materials Lab, MNIT and DST Government of India for providing funds. And thank you all for patiently listening.